Hello. I'd like to welcome everyone today to another ADA Spotlight the Science, where we meet our amazing ADA funded researchers. So my name is Marlon Pragnell. I'm Vice President of Research and Science at the American Diabetes Association. And it's really a pleasure and a privilege to be able to speak today to Dr. Sarah Tishkoff. Dr. Tishkoff is a professor at the University of Pennsylvania, and she's in the Departments of Genetics and Biology. And she's received one of our premier awards, the Pathway Award, in 2019, and she's working to study genetic risk factors for adult onset diabetes in populations of African ancestry. And looking through her bio, she's actually been her research has been discussed or featured in many articles in the New York Times and the Washington Post, NPR, and BBC. So it's a real privilege to speak to Dr. Tishkoff. Welcome.
Thank you. It's my pleasure to be here and tell you a little bit about my research.
I think it's fascinating research. I know that you span both Africa and also to the States in terms of understanding the genetic factors leading towards disease. Can you tell us a bit more about the work you're doing?
Sure. Well, first of all, my background is in anthropology, human genetics, and evolutionary biology. And one of the exciting things about this Pathway Award is that it's for people like me who don't necessarily have a training in diabetes research. This is new for me. This is a new direction, and that funding is enabling me to go in this new direction. And so just to tell you a bit about what got me into this and sort of the general type of research that we do, we are studying genetic variation, mainly in sub-Saharan African populations. We integrate that with what we call phenotypic variation. That just means whatever trait you're looking at. Sometimes it's normal variation. It could be lactose tolerance, skin color, height, weight. And then there's also things like resistance to infectious disease and cardiometabolic types of traits. So our research in the lab integrates fieldwork in Africa together with African collaborators and partner institutions. We do what we call functional genomics research, and then we have a lot of computational research in the lab. So we integrate those three. So one thing that we do is we've been doing fieldwork now for over 20 years, mainly studying minority populations in Africa who are practicing more traditional lifestyles. So we've been doing work in, my gosh, many countries: Sudan, Ethiopia, Kenya, Tanzania, Cameroon, Nigeria at one point, and Botswana. I might be missing one somewhere in there. And so these populations tend to live in very remote areas. They can be very challenging to get to. And they have no electricity there, typically. So we basically bring all our supplies, we bring all the lab supplies, and we set up a lab in the bush, so to speak. We have to use a generator. And a lot of work went into just perfecting how to do that and how to preserve samples under those conditions. And these populations most of them have never been included in any genetic studies, and certainly very, very limited biomedical research in these populations. But what makes them so informative is that we see the most genetic diversity in the world in Africa. And that's because all modern humans originated in Africa around 300,000 years ago. Somewhere around, say, 80,000 years ago, relatively small numbers of people migrated out of Africa, giving rise to populations across the world. But because of that history, populations have been in Africa the longest. They've maintained relatively big population sizes over a longer period. And so they just maintain a lot of variation. And because they have adapted to many different environments and different diets, they also have a lot of genetic diversity and a lot of this phenotypic diversity, variation for a number of different traits, including risk for disease. So part of this is the challenge of even doing this research. And one thing we're particularly careful about is to do this research in an ethical manner. So we want to make sure that we are working closely with African collaborators and people from the local communities going through very extensive ethical review at all levels, not just in the US, but also in each country. And then we spend a long time just going to the communities, talking to them, explaining what we're doing, why are we doing it, what are the benefits, if any, what are the risks, if any, addressing questions, addressing concerns. And then only when we have community consent, we ultimately have to get individual consent. And I think the other thing is that we involve the community members actually even in collecting the research, and we do some even training of local people and things like that. So, so in the field, we're able to collect, for example, DNA and cell lines from blood, and we can do some measurements. We look at, for example, blood sugar in everybody, baseline blood sugar. We've done, we look at blood pressure and some different cardio heart function related traits, and we can look at infectious disease status and so on. And then when we come back to the lab, at this point, we're actually doing whole genome sequencing. So the technology's gotten to a point where the costs have come down that we can actually sequence entire genomes. And this is actually part of a collaboration with, it's called the Top Med Initiative at NIH. And they are doing a lot of the sequencing of genomes, and we're part of that. And then you can, from that genome sequence, one is just characterize the nature of the variation that they have. The second thing you can do is look at genomic footprints of natural selection. So there are certain signatures, and when I say natural selection, I mean, I'll just give as an example that many people are familiar with sickle cell disease. So sickle cell disease is caused by mutations in the beta globin gene. If you were to have 2 copies of the sickle mutation that causes sickling of the red blood cells, and it's extremely detrimental. In fact, results in death by typically around age 5 if left untreated. But people who have 1 copy of the sickle mutation and 1 normal copy are actually protected from malaria. And malaria kills millions of people every year. So because of that, this variant is maintained in the population for at a relatively high level. And that's why we see a relatively larger number of people of West African ancestry having sickle cell disease.
Wow.
But that same phenomenon might be happening that's causing diabetes as well. So yeah, something that's called the thrifty gene hypothesis, for example, is that of course, we would be adapted to the environment over the past, say, thousands of years. And in certain regions, traditionally, actually, all of us until about 10,000 years ago would have been practicing a hunting and gathering lifestyle. Our ancestors would have been practicing a hunting and gathering lifestyle. In the past 10,000 years, there was the development of agriculture and farming. And there was also domestication of animals. And so you get like dairying, for example. And so when the diet changes and the environment changes, there are different selective forces, and there may be genetic variants that are adaptive in that environment. Now, if in the past people are going through periods of starvation, for example, then maybe it's adaptive if you have some genetic variants that are going to help you store fat. But now they're coming, people are in the US environment where there's such an excess of food, and now it's detrimental. And that could result in obesity and diabetes, for example.
Fascinating. The idea that you could have some genes or some adaptation to your environment, and as you mentioned, Africa, such diversity there, in terms of people living in locations and over many generations adapting to their specific context, their environment. So, one location, for example, protection against malaria or so. So that's fascinating. So obviously when we don't need to be protected against malaria here in the States. So obviously then people bring over when African Americans here in the States harbor these various genes that are, sickle cell is probably not, we don't need sickle cell gene to protect ourselves from malaria. But you mentioned also too then that some of these genes then can actually, where they would be helpful in an ancestral environment, could be quite detrimental. in this environment.
Yeah, exactly. And so I was saying then one of the ways you can find these variants is you look for these genomic footprints. They leave a typical signature. When something is adaptive, it gives you some protection from something, and you're more likely to have children, and your children are more likely to have children, and it rises rapidly in frequency. And that actually leaves a genomic footprint. We can actually try to find that. And then the other thing we can do is integrate the genomic data with information about traits like cardiometabolic traits and blood sugar levels and things like that. And so that can help us to identify functionally important variation that's playing a role in cardiometabolic traits. But one of the things that motivated me to apply for this Pathway Award was actually an observation. So these groups that we study, as I said, they practice more traditional lifestyles. Some of them are hunters and they're doing hunting and gathering. Some are farmers. Some are pastoralists, meaning that they have cows and they practice dairying, and then everything sort of in between. And but they don't have a lot of access to grocery stores. They're not going shopping. They're, and so we rarely see diabetes. We did not see it very often, and we rarely saw hypertension. But when we were in Cameroon, one of the ethnic groups that we were studying, called the Fulani, we saw an unusual high prevalence of an adult onset diabetes and hypertension. And that just stood out to us because we don't typically see this in our studies. And the average BMI in that population is about 19. They're not overweight.
Yeah.
They don't have the typical risk factors. Why are they getting diabetes? Why are they getting hypertension? And that was like the mystery that we wanted to solve. Now, this particular group, their ancestry is they actually originated, we think, from North Africa, like a Berber ancestry, maybe from East Africa, and they have cattle. And when you have cattle, you have to migrate. So they migrated across the Sahel from across Central Africa. And when they migrated, they interbred with people who were traditionally farmers. And I could tell you that from our prior studies looking at genetic variation in Africa and in African Americans, we found that most African American ancestry derives from these people who are like the farmer ancestry, let's put it that way. They speak what we call Bantu languages, and we see predominantly that ancestry. But we see up to 14% ancestry similar to the Fulani. Okay, so we will see this in US populations who originated from Africa. Now, the other really unusual thing about the Fulani is that they have relative resistance to malaria compared to other ethnic groups living, say, in the same exact area. And they don't have sickle cell mutation. They don't have any of the known variants that play a role in resistance to malaria. So it's a big mystery. What's going on here? There's some suggestions that it might be an altered inflammatory immune response. And so our hypothesis when we went into this was, what if they have some genetic variants that are protective against infectious disease, but they may also predispose you to adult onset diabetes and hypertension. And if we could understand that mechanism and find the genetic variants that are playing a role in this, that can really be important, possibly for understanding risk factors for adult onset or certain types of adult onset diabetes, because it is known that while obesity is highly correlated with type 2 diabetes, there are some people who are not obese, and yet they're getting type 2 diabetes. Maybe this will give us some clue about why that is. And could it actually be that immune function is somehow playing a role?
Fascinating. So then this protection, this immune response, this inflammation that would have been valuable in an ancestral African context to protect against disease, you're having this now being in the Western context being detrimental. Then, in a sense, more like increasing the risk of diabetes or
Correct. Yeah, because we know that inflammation, I mean, it's been observed that inflammation certainly is correlated with things like hypertension and diabetes. And some people say it could possibly be causal. And so that's something that we want to determine. And then the other thing more broadly is that we have data from thousands and thousands of ethnically diverse Africans from many geographic regions of Africa, representing some of the most diversity in the entire world. And so we are also collecting cardiometabolic traits. So I mentioned we look at baseline blood sugar levels. When we go back, we're going to go back to the field. We want to do glucose challenge test. And then we also have information, very detailed information about insulin levels and about lipid levels and about all kinds of biomarkers in the blood that are associated with risk for diabetes, for example. And we can then look at the correlation between the genetic variation and these biomarkers and try to identify what are the variants, genetic variants that might be correlated with that or might be causing variation for these traits. And again, that could be very helpful for potentially identifying therapeutics that would be more effective in all ethnic groups.
Yeah.
And the other reason it's important is because nowadays, when people do these studies, they're called genome-wide association studies. So what they do is they look at genetic variation across the genome in a population of individuals for whom they have, let's say you're looking at risk for diabetes. And you look for the association between these variable genetic sites and disease. And if you see an association, then it means that those genetic markers must be physically very close to the mutations that are causal or playing a role in risk for disease. It's a way to help map where they are. Almost all the large studies have been done in people of European ancestry, most genome-wide association studies. And what people are doing now is calculating something called a genetic risk score. Sometimes it's called a polygenic risk score, or PRS for short. And what they do is they add up all the associations. So if you find a number of different variants that are associated, let's say for risk for diabetes, and you want to look at a patient who was not part of that original study, and you want to know what's that patient's risk for disease, you add up the effect of all those different variants. But what people have shown is that this works really well if your patient is of, say, European ancestry, and that's where your original study was done. But it does terribly when people are from other ancestries, and it does the worst in people of African ancestry.
Yeah.
So we need to do a better job with figuring out better genetic risk scores because these are being implemented in the clinic. They're being used to know who's at high risk for disease and doing preventative work or therapeutics or so on.
For sure. This is in a sense your work is also addressing a health disparity as well, because if you're looking trying to identify something in a population that's very different from the population you've studied studied before, then you're not being very accurate. But this really suggests precision medicine as well, where
Absolutely.
you have people, you understand the genetic makeup and you are better able to deliver the right therapy to the right patient at the right time.
Exactly. But we're only going to discover that if we have a more diverse set of individuals in which we're doing this research. So, I mean, we also know that people vary in terms of variants at genes that play a role in drug response and drug metabolism. And if you're only doing this research in Europeans, they may not be as effective, or there might be actually negative side effects. That's a bigger problem sometimes.
Sorry, go ahead. One of the exciting things here is the potential for identifying new genes and targets, developing drug approaches. And so if you understand the genetic cause of disease, and if you understand for example, there's a molecular pathway, then you could actually identify potential drugs or therapies to better treat people. So it could lead to new drugs ultimately in the future.
That's true. Yeah.
But sorry, go ahead. You were mentioning what you were saying.
I was actually just looking at the comment here.
Let's look
so
at
somebody was asking about what was lacking in the past research initiatives that were unable to provide supportive evidence in favor of the thrifty gene hypothesis for diabetes and still remain elusive. What should be the way forward in this field? Yeah, you're right. Actually, this is, I think, Swapan Kumar Das. Thank you for that question. You're absolutely right. And actually, it's been somewhat controversial. People have debated whether this thrifty gene hypothesis is true and can it actually explain diabetes. But in terms of what we need more research in ethnically diverse populations. Now, for example, there has been some really interesting research done in Samoan populations where they identified a gene that showed the signature of natural selection that I was talking about and was associated with BMI in that population. But the problem is when you're trying to prove, is it caused by the thrifty, this whole thrifty gene hypothesis, you have to know what the selective force was. And honestly, that's sometimes hard to know. But I think when you look at enough populations that are adapting to different diets and environments, you can start to piece that together. But until we study these different populations, we're never going to know. But there's also this other hypothesis I raised. Maybe it has to do with immunity. Maybe it has to do with exposure to infectious disease and that alterations in the inflammatory immune response can play a role in risk for disease. And that's something else that we're trying to test.
That's fascinating. I think this work that you on human genetics, such tremendous potential to basically deliver better therapies, better treatments, and so for people. Tell me, you said you did anthropology. So what got you into this? What got you into the study of anthropology? It's such a fascinating field.
I think I can trace it back to reading a book by Margaret Mead in high school. And she was a famous social anthropologist. And actually, she was studying like Samoan populations. And I said to myself, I want to do field work. That's what I want to do. I thought I'd be a social anthropologist, cultural anthropologist. I went to UC Berkeley, started majoring in anthropology. But at Berkeley at that time, there was a brilliant biochemist, researcher, molecular biologist named Dr. Alan Wilson. He's considered the founder of molecular anthropology. So using genetics, for example, to reconstruct our past. And some of his academic offspring were also there at Berkeley, and I was taking classes with them, and I even ended up working in their research labs. And that just got me fascinated in this whole field. But I realized that I needed a stronger foundation also in genetics. So I double majored in anthropology and genetics, and I worked in several genetics research labs. And then for graduate school, went to Yale, where Professor Kenneth Kidd was one of just a couple of people in the country who had collected cell lines from populations across the world, giving me an opportunity to study diversity globally. But the only populations they had from Africa, there were just two. They are Central African hunter-gatherers. They were being used as representative Africans. They're not representative of the diversity in Africa, but nothing existed. There were just so few samples. So I started writing to researchers who did have some African samples and started looking at the patterns of genetic diversity. And what I saw was just striking. There was so much diversity amongst ethnic groups in Africa, even in a small region. You could look in like a 100-kilometer region and you will see so much genetic diversity within and between groups that I just didn't see in the European populations or East Asian populations. And that got me fascinated. Why do we see all that diversity? What could cause that? And then for my postdoctoral research, I went to work with Andy Clark to study population genetics. But before I even made it to his lab, I did a short stint at the University of the Witwatersrand with Dr. Trevor Jenkins, who was one of the leaders in studying African genetics. And again, that just inspired me even further. And then once I got my own lab, I could finally go to the field and fulfill my dreams, which was to actually do field work.
Fascinating. I mean, when I read your work, so it's amazing. You can do research here in the States, but then go to fascinating locations. Leveraging all the genetic gene pools in the different locations. Absolutely fascinating the way you bring us into the present day with trying to understand the genetic risk for diabetes and other diseases. So, I think we've actually run over time. This has been so interesting, and it's such a pleasure to chat with you. And also, just want to thank you for doing such phenomenal research and such interesting research. And it's great to have you as one of our American Diabetes Association grant recipients. Thank you so much.
Yeah. Well, I have to say thank you so much. Thanks to the ADA and the people who've supported the Pathway program, because I wouldn't be doing this without that. It really helped me move into a new direction, and so glad that I was able to share a little bit of my work with you today.
Thanks so much. Fascinating. Good.
Take care.
Take care.